for inviting me here. I'm very glad to be here. I participated in a similar event 4 or 5 years ago, and I have really fond memories of this event. I am now in Princeton University, but definitely going back to Ben-Gurion University at the end of this year. And I will tell you about long research that have been done in my lab in Israel that is focused on face processing and mostly struggling with face processing. And before going into the science, I also want to share some kind of a sort of more personal perspective about my sort of involvement in the Negev and partly why I'm there. So this is sort of my own family history. This is my grandfather and my grandmother, with my mother and her sister and my great-grandmother. And my grandparents were actually among the founders of a kibbutz in the Negev. It's called Gat, and this is what it looked like many years ago, and this is how it looks like from today. And actually, my family is now in this kibbutz. This is where we live, and this is where we've been living since I arrived to Ben-Gurion in 2006. So it's kind of a personal sort of involvement with a lot of history attached. And so going to the research, so faces, like why do we actually study faces? What's so interesting about faces, and why are they important? So we know that at the behavioral level, actually face perception is probably one of the most developed visual perceptual skill that we have as human. And when we think about faces, we're actually quite similar to each other. So we'd like to think of ourselves as being sort of special, but actually we all have eyes and a nose and a mouth, and it's sort of located in the same place. And so it's a really difficult sort of category of visual stimuli to deal with, because it's very homogeneous. So yeah, and as I mentioned, all the elements are kind of organized in the same way, and this organization is very important actually for recognition. But faces also convey a lot of information. So we know a lot when we look at a person. We know their age, their gender, the emotion that the face portrays, and the kind of research that we're most focused on is knowing who's that person, so knowing the identity. And we also know that faces are actually processed in a very unique fashion, which is quite different from the way our brain process other types of visual information. And we relate to that kind of processing as holistic processing, which means that basically we're not so focused on understanding the little details that are in the face, but we're sort of capturing the entirety or the totality of the face. And we often don't pay attention to it. And sometimes, like, if you think about a person that you know really well and try to sort of figure out the details of their appearance, sometimes you don't even know it. And sometimes you don't even know, like, the colors of their eyes, even very trivial things. So, and I also like to show these portraits that are done by the Israeli artist Hanoch Piven, who creates these beautiful caricatures, which sort of captures very nicely how artists understand things that we struggle with. So you can see how with very, very little information and minimal details, he actually managed to create these portraits that are very sort of clear to us. So I hope that all of you can recognize these faces. And if not, come see me after the talk, because we're always looking for more participants to our studies. But interestingly, faces are really very sort of immune, or our face recognition abilities are sort of immune to very drastic changes to the viewing conditions. And these are kind of blurred versions of famous people, which I also hope you can recognize. It's like Woody Allen and Clinton and Princess Diana and Prince Charles. So these are really blurred versions, but still we are able to recognize them successfully. And interestingly, going back to the developmental aspect that Ilan mentions, this is actually how very young babies see faces. So all they have is like blurred versions of faces because their visual acuity is poor. So it's interesting that our face recognition system develops in a way that allows us to sort of capture such important information even in these very minimal images. But yet, our face recognition abilities are very sensitive to other manipulations that are computationally much simpler. And one important one is inversion. So this is, again, going back to art. This is a famous painting in which presumably all of you just see sort of a ball with some vegetables in it. But if I only just invert this image and really doing nothing else, I hope that you can now see that there's a face in that image. So, and you haven't seen the face when it was sort of upside down. So our face recognition system is really very sensitive to the orientation of the face because we're really, most of our life, are sort of trained on looking at upright faces, and the system became tuned to understanding and recognizing those faces and not their inverted version. And faces also have very sort of unique and social significance for humans in terms of sort of conveying information about emotion and eye gaze, and even very abstract sort of concepts such as trustworthiness. So we can look at someone and sort of know whether we trust that person or not without sort of understanding what is it about that person. And again, this goes to the research that Ilan was mentioning about autism, and we know that often children with autism have difficulties with sort of looking or paying attention to faces, and they're not looking at faces, and of course they also have some social difficulties in interacting with human and understanding social interactions. So going to the neuroscience of faces, and indeed face perception is also sort of unique from a neuroscience perspective. And in Ben-Gurion, and of course in other places in the world, we can study face representation or the way the brain processes faces using MRI. So this is our own sort of MRI in Soroka. And we can understand the sort of the structure of the brain, but we can also use this amazing machine to understand the function. So what is it that the underlying neurons are actually doing while we look at faces? And over the last several years, there's sort of a growing consensus among researchers that faces are really sort of being processed by a distributed network in the brain and not by specific regions. And what we can see here is an image with data from many participants who basically viewed different images in the scanner, and we can see in orange and in yellow the regions that are activated when these people see faces. So we can see them here when the brain is shown laterally, but we can also see these regions when we look at the sort of bottom part of the brain when it's rotated and we look at it sort of from the ventral aspect. And the research suggests that there is sort of a core system for face recognition that is focused on more posterior parts of the brain and deals with the visual aspect, the sort of purely visual aspects of face recognition. But there's also an extended system that involves regions that are related to memory of the faces and associated biographical knowledge and things like names and emotions to the faces. And so because it's a network, we really need to have some tools to study this from a network perspective. And this is what we've been doing over the last several years in my lab. And so we rely on tools that come from basically computer science and mathematics, and this is a whole field that is called the science of networks. And we know that networks can describe many complex patterns and phenomena, such as social interactions and disease spread and gene expression. And actually, thanks to the work of several important people, one of them is Olaf Sporns, who was a collaborator of mine in Indiana University, we now understand that the brain can also be described as a complex network. And so our approach is to study the topology, sort of the organization, the underlying organization of the normal and the impaired face network using this approach. So basically what we want to do is to take these images showing sort of single regions that are activated when people look at faces and generate networks that describe the way these different regions are actually interacting and how this happens in a normal brain and how this might be impaired in individuals who cannot recognize faces. And so the basic sort of building blocks for every networks are the nodes and the edges. And we can see sort of a simplification of that here. And again, as I mentioned, our goal is not to just describe those elements, but describe the interactions between these elements and the topology, the underlying connectivity. And so just to sort of give you an example of what I mean by topology, I want to show you a sort of real-world example of topology. And this is a study that was done on social networks. So this is Twitter. And you can, again, apply these tools to many, many networks. And what is shown here is that if you examine the properties of specific networks on Twitter, you can see very interesting characteristics. One of them is shown here. This is called the polarized crowd network map, which basically means that it's composed of sort of two big communities. One here happens to be a group that is sort of a more liberal, and the other is a more conservative group. And the interesting thing is that you see that sort of people within each group talk a lot to each other, but then there are only very few people that actually convey information between these two groups. So there are kind of two segregated groups without much integration. But then there are different other groups on Twitter that you can find, and this is a different description. This is a tight crowd network map, which means that all these people talk to each other. So there are no two sort of isolated groups as before, but now there's sort of much more communication between everyone. But some people within this group, or some members, are sort of more important than others, and they serve like hubs. So many, many people connect through them, and if we take this hub away, then the network would change in a very drastic way. So I hope you sort of get that idea of how to apply mathematical tools to describe these complex networks. And this is what we've been doing, again, for studying the normal face processing system, but also to understand what happens when its function is abnormal. So the first study that I want to describe to you was done by a past graduate student in my lab, Gideon Rosenthal, now Dr. Gideon Rosenthal, in collaboration with Professor Olaf Sporn from Indiana University, who I just visited before coming here. And in this first study, this was sort of our first attempt to describe the normal face network, and we wanted to use a very robust behavioral phenomena to do that. And as I showed you, we are very sensitive to inversion of faces. So all of us are actually impaired in recognition when we see inverted faces, even though we are very familiar with the face. So you can't get around this phenomena, really. And you can see the image over and over again, and you will always be kind of slower in responding to this image compared to its sort of normal upright version. So we wanted to take this phenomena and study what is the sort of neural representation of this effect, but not just going to specific regions and examining whether they respond more or less to the upright versus the inverted faces, but we wanted to see it sort of from a network perspective and develop this kind of fingerprint of the inversion effect, or the connectivity fingerprint for the inversion effect. So the results. So first of all, we always have to look at the behavioral level, right? Because if the people in the scanner do not show the effects that we want them to show, then we need to sort of rethink our approach. So just to sort of quickly reassure you, the participants in this particular study showed the expected effect of being less accurate when they needed to judge inverted compared to upright faces, and they were also sort of slower in responding to upright compared to inverted faces as we want them to be. And now the question is, what does this look like sort of in terms of the network, in terms of the connectivity? So what we did is to generate these nodes and edges, and the nodes here are depicted in the different circles, and the nodes that are depicted in orange are those regions that were face selective, so where we think that there are neurons who are responding more to faces. And the green nodes and the blue nodes, mostly the green, are those that responded to other stimuli, so to other visual stimuli that the participants saw in the scanner. And what we can see here in the red lines are the connections, sort of the edges that connect different nodes. And we can see that when participants viewed upright faces, there was a lot of connectivity between face regions. So the nodes that were selective for faces, that responded more to faces, were also more connected to each other when the participants viewed faces. And then the question is, what happened when we do the opposite comparison? So are there different connections in the brain when we see inverted faces compared to upright faces? And the answer is yes, because we now look at this network of participants viewing inverted compared to upright faces, and we can see that actually the network changed drastically. So it's really not something that is very subtle, involving one sort of connection or the other connection, but it's really sort of a whole brain-wise difference that we can see here. And the other interesting point is that now there are the sort of orange nodes that were so significant in the previous image that I showed you are kind of almost gone. And what we see here is that there's stronger involvement of these green nodes that are actually representing areas that responded not to faces but actually to objects. And these areas are now strongly sort of interacting with each other. So this suggests to us that the way that our brain represents inverted faces, it's really more akin to the way we process objects. So it's not longer sort of a proper face in its upright orientation to which our brains were sort of tuned to and learned to recognize, but if an inverted face is being processed in a more kind of object-like fashion. And again, just to sort of convince you that this is a widespread phenomena across many, many regions, I'm just sort of flipping between these two images just to show you how different these patterns are. So basically using this network level approach allowed us to generate this signature of the face inversion effect. And it's interesting because it's really sort of a simple physical transformation of the stimulus itself, but it induces this very dramatic functional reorganization across many, many brain regions. And it's only due to this network approach that we developed that we could actually see this widespread phenomena. So now I'd like to move to our second study that involved using this same approach. But now we wanted to focus on individuals who are actually impaired at face recognition. And it's a very interesting phenomena that is called congenital prosopagnosia. So it has a difficult name. But these individuals actually exhibit face processing impairments from birth, as far as we can tell. So we know that Some people might suffer a brain lesion due to some brain trauma, and then their face recognition might be impaired due to that lesion. But this is a different group. This is a group of individuals who are otherwise completely normal. They do not have any sort of neurological condition. It's just that face processing is hugely difficult for them. And one such individual is an American painter, Chuck Kloss, who you may be familiar with. And interestingly enough, he likes to paint faces. So it's an interesting choice for someone who finds it so difficult to recognize faces. And I like to show you this short clip from an interview with him.
In the face blindness, the name is how do you say it?
Prosopagnosia.
Yeah. Now that means that you can't recognize faces. You have trouble.
It's a sliding scale.
Yeah.
I have a great deal of difficulty recognizing faces, especially if I haven't. If I've just met somebody, it's hopeless. I will never remember them again, unless it's reinforced over and over and over. And even people that I know very well, if I haven't seen them for a while, it's like a bucket with a hole in it. Information is coming in, but it's pouring out the bottom just as fast. And I'm often losing information.
But you are known for portraits of faces.
I was driven to make them. I'm absolutely positive.
That's what did it.
Because in real life, if you move your head a half an inch, to me it's a whole new face I've never seen before. But if we flatten it out, I have, and I take photographs, I work from photographs, and I make flat things called paintings and prints. I have virtual photographic memory for anything that's flat. So it's not an accident that I only do images of people who matter to me, family, friends, other artists. There are no commissioned portraits.
So it's always interesting for me to hear the description of these people about the way they see faces and the way they process faces, and it provides us a lot of insight about the way these stimuli are processed for them. And again, as I mentioned, it's related to the phenomena that is called acquired prosopagnosia, which is caused by brain lesion. But for these people, there's no cortical lesion. There's nothing sort of evident that is obvious in their brain structure or function. And they all had normal opportunity to acquire face processing. And interestingly, again, going to genetics, it turns to run in families. So we often see one family member who tells us a lot about other family members who suffer from this. And the prevalence is quite high. So it sort of ranges between 1% and 2% of the population, which is quite striking. And I put these inverted faces here. This is taken from a journal article that was written about this work in Israel. And again, I just to sort of flash it to you, just to make it clear how these individuals might experience faces. So you by now you know that it's hard to recognize inverted faces, and for them it's not that they see faces as upside down, but the sort of the difficulty of distinguishing between different faces is somewhat similar to what we experience when we see these inverted faces. And I hope you can recognize at least some of these images. Some are sort of famous also here in the States. You can imagine like going around the world and not being able to recognize sort of people around you, even if they're very, very close to you. And so when we started this project many years ago, actually, we thought it's going to be like the shortest project ever, because we were just, planning to scan these individuals in fMRI, and they're not recognizing faces, and we know that there are regions in the brain which are important for recognizing faces, and we thought, we'll just find that these regions are abnormal in their brains and just be done with it. But interestingly, this is not what happened. And so these are, again, the brains of normal individuals, normal in terms of their face recognition. And this is a group of prosopagnosic individuals who were scanned under the same condition. And much to our surprise, in this initial study and in our studies over and over again, was that their face recognition regions were completely normal. So what do we do with it? And this is where our sort of network approach became very important, because by now you know that there are regions, but there are also the connections between these regions that are actually critical for the processing of the information. And so our goal here was to examine the functional connectivity within each of the core and the extended face network, and importantly, the interaction between these two networks. And this was a collaboration, again, led by my former student, Gideon Rosenthal, and another student in the lab, Michal Tenzer, and colleagues from Princeton University, and Professor Marlene Berman from Carnegie Mellon University, who has been a great colleague for many, many years for me, and also to Ilan Dinstein. And so looking at their connectivity, what I'm showing you here are sort of similar images as before, but now the comparison is not between the conditions, like the upright and the inverted faces. Now the comparison is between groups. So what we're looking at here, the red lines are the edges, the connections that are stronger in the control group, in the normal group, compared to the group of the people that have difficulties in face processing. And we can see, again, that there are all these orange nodes that appear when the controls are looking at the faces. But importantly, there's this big node here in the region that is called the anterior temporal cortex. So it's the temporal lobe is the one here, and it's the sort of anterior part of it. And there's a region here that, again, based on the calculation that we were able to do, really sort of behaves like a hub. So it's an important region. It connects many, many other regions that are related to face processing. And it suggests that if something wrong with this hub, then the network is going to be very, very different. And interestingly, this is exactly what we found in the individuals with congenital prosopagnosia. So these are the networks of our prosopagnosic individuals compared to the network of the controls. So the lines here represent the edges, the connections that are actually stronger in the prosopagnosic group compared to the controls. And the striking thing that we can see here is that this node, this important hub in the anterior temporal cortex, is completely missing from the network. But it's not because it doesn't exist in their brain, right? This region is there, it looks completely normal, but its connectivity is sort of absent from the face network. And instead, what we see is sort of vast connectivity between more posterior regions in more sort of visual parts of their brain. And these regions, as you can see by the color, are not even sort of mostly face-related regions, but these are regions that process sort of general visual information, so are related to object recognition or scene recognition. So again, this is kind of akin to what I was told you about what the brain looks like when we examine inverted faces, suggesting that, again, these individuals actually look at faces using mechanisms that are more related to general object processing and not so specific to face processing. So again, using this perspective allowed us to find this and to view in a sort of a very interesting perspective where the differences are between the two groups. And again, just to sort of convince you that this is a striking whole brain phenomena, I'm again flipping between these two images, and you can see how different these networks are across the two groups. Okay. So again, just to sort of summarize, I've shown you that faces are special from both a behavioral and neural perspective, and I hope I convinced you that fMRI can be used to discover brain connectivity that is unique for face processing compared to the processing of other stimuli. but it's also a very useful approach to understand impairments in face processing and how they sort of being reflected in the differential connectivity in the brain. And the other important point here is that these tools that have been developed uniquely for face processing can now be applied for other conditions. So they're very general in that sense. And we can study other phenomena, other cognitive disabilities or neurodevelopmental disorders using the same sort of mathematical computational tools. And in terms of general implications for this research, so Google Glasses could be one application, definitely, that could be very useful. But sort of from a basic science approach, and this is where my heart is, it's very important for us to understand this multifaceted phenomena of face perception, again, both behaviorally but also neurally. And this unique phenomena of congenital prosopagnosia provided us with a very sort of special window to that topic. Of course, it also has important social implications. Again, as I mentioned, individuals with autism often exhibit difficulties in face processing, so it's important to understand how face processing is sort of being computed in the normal brain and maybe in people with autism or people suffering from other difficulty due to brain trauma. Again, going back to developmental perspective, of course, congenital prosopagnosia is a neurodevelopmental disorder because we hear stories from people that have early memories of their difficulties and how they struggle with their difficulties. And you can imagine a kid in kindergarten not knowing that there is such a thing as prosopagnosia, obviously, but struggling to recognize their peers or their caregiver. This could be very traumatic. And again, we hear stories from the participants that come to the lab about their early memories and sort of the most kind of basic thing, that the most traumatic thing that they describe is like being separated from their parents in a big crowd because they were always worried that they might not be able to find them. So this is scary. And of course, sort of from a more applicative point of view, we're interested in developing different aids for face perception, either behavioral aids, cognitive aids, but also computational or automated tools for face perception, such as Google Glasses, but also other applications. And I'd just like to end by thanking all the people that did the hard work and were involved in this over the years. First of all, the students in the lab, Gidon Rosenthal, Gidon Levakov, Michal Tenzer, Nili Weiss, Karen Lessinger, and Carmel Sofer, who all took part in various aspects of these projects. And Olaf Sporns from Indiana, who's a great collaborator, Marlene Berman from Carnegie Mellon University, Uri Hasson and Erez Simoni from Princeton University, and of course the funding agencies. And thank you very much for listening.